Pathophysiology of gestational diabetes. The precise mechanisms underlying gestational diabetes remain unknown. The hallmark of GDM is increased insulin resistance. Pregnancy hormones and other factors are thought to interfere with the action of insulin as it binds to the insulin receptor. The interference probably occurs at the level of the cell signaling pathway beyond the insulin receptor. Since insulin promotes the entry of glucose into most cells, insulin resistance prevents glucose from entering the cells properly. As a result, glucose remains in the bloodstream, where glucose levels rise. More insulin is needed to overcome this resistance. About 1.5 to 2.5 times more insulin is produced than in a normal pregnancy. Insulin resistance is a normal phenomenon emerging in the second trimester of pregnancy, which in cases of GDM progresses thereafter to levels seen in a non-pregnant person with type 2 diabetes. It is thought to secure glucose supply to the growing fetus. Women with GDM have an insulin resistance that they cannot compensate for with increased production in the beta cells of the pancreas. Placental hormones, and to a lesser extent increased fat deposits during pregnancy, seem to mediate insulin resistance during pregnancy. Cortisol and progesterone are the main culprits, but human placental lactogen, prolactin, and estradiol contribute, too. Multivariate stepwise regression analysis reveals that, in combination with other placental hormones, leptin, tumor necrosis factor alpha, and resistin are involved in the decrease in insulin sensitivity occurring during pregnancy, with tumor necrosis factor alpha named as the strongest independent predictor of insulin sensitivity in pregnancy. An inverse correlation with the changes in insulin sensitivity from the time before conception through late gestation accounts for about half of the variance in the decrease in insulin sensitivity during gestation. In other words, low levels or alteration of TNF-alpha factors corresponds with a greater chance of, or predisposition to, insulin resistance or sensitivity. Gabby Stephen G., 6th edition, page 890. It is unclear why some women are unable to balance insulin needs and develop GDM. However, a number of explanations have been given, similar to those in type 2 diabetes, autoimmunity, single gene mutations, obesity, along with other mechanisms. Though the clinical presentation of gestational diabetes is well characterized, the biochemical mechanism behind the disease is not well known. One proposed biochemical mechanism involves insulin-producing beta cell adaptation controlled by the HGF/c-Met signaling pathway. Beta cell adaptation refers to the change that pancreatic islet cells undergo during pregnancy in response to maternal hormones in order to compensate for the increased physiological needs of mother and baby. These changes in the beta cells cause increased insulin secretion as a result of increased beta cell proliferation. HGF/c-Met has also been implicated in beta cell regeneration, which suggests that HGF/c-Met may help increase beta cell mass in order to compensate for insulin needs during pregnancy. Recent studies support that loss of HGF/c-Met signaling results in aberrant beta cell adaptation. c-Met is a receptor tyrosine kinase, RTK, that is activated by its ligand, hepatocyte growth factor, HGF, and is involved in the activation of several cellular processes. When HGF binds c-Met, the receptor homodimerizes and self-phosphorylates to form an SH2 recognition domain. The downstream pathways activated include common signaling molecules such as RAS and MAPK, which affect cell motility, and cell cycle progression. Studies have shown that HGF is an important signaling molecule in stress-related situations where more insulin is needed. Pregnancy causes increased insulin resistance and so a higher insulin demand. The beta cells must compensate for this by either increasing insulin production or proliferating. If neither of the processes occur, then markers for gestational diabetes are observed. It has been observed that pregnancy increases HGF levels, showing a correlation that suggests a connection between the signaling pathway and increased insulin needs. In fact, when no signaling is present, gestational diabetes is more likely to occur. The exact mechanism of HGF/c-Met regulated beta cell adaptation is not yet known, but there are several hypotheses about how the signaling molecules contribute to insulin levels during pregnancy. c-Met may interact with FOXM1, a molecule important in the cell cycle, as FOXM1 levels decrease when c-Met is not present. Additionally, c-Met may interact with P27 as the protein levels increase with c-Met is not present. Another hypothesis says that c-Met may control beta cell apoptosis because a lack of c-Met increases cell death, but the signaling mechanisms have not been elucidated. Although the mechanism of HGF/c-Met control of gestational diabetes is not yet well understood, there is a strong correlation between the signaling pathway and the inability to produce an adequate amount of insulin during pregnancy, and thus it may be the target for future diabetic therapies. Because glucose travels across the placenta, through diffusion facilitated by GLUT1 carrier, which is located in the syncytiotrophoblast on both the microvillus and basal membranes, these membranes may be the rate-limiting step in placental glucose transport. There is a 2 to 3-fold increase in the expression of syncytiotrophoblast glucose transporters with advancing gestation. Finally, the role of GLUT3/GLUT4 transport remains speculative. If the untreated gestational diabetes fetus is exposed to consistently higher glucose levels, this leads to increased fetal levels of insulin. Insulin itself cannot cross the placenta. The growth-stimulating effects of insulin can lead to excessive growth in a large body, macrosomia. After birth, the high glucose environment disappears, leaving these newborns with ongoing high insulin production and susceptibility to low blood glucose levels, hypoglycemia.